The light reactions occur in photosystems. Photosystems are complexes of proteins and pigments embedded in the thylakoid membrane. So putting it back into the chloroplast. And every photosystem has two components, the light harvesting complexes and a reaction center. These are also sometimes called the antenna complexes because their role is to catch light. So light will shine down and it will hit the light harvesting complexes, and they pass that energy down to the reaction center. So light harvesting complexes collect the light. That's where they get the name antenna too. They pass the energy in the light to the reaction center, which is the second part. In the reaction center, there's a special chlorophyll molecule that will absorb the energy by getting an excited electron. So the excited electron occurs in the reaction center, and that excited electron is then captured by something called the primary electron acceptor. So that's the basic process in the photosystems. There's two types of photosystems, photosystem 2 and photosystem 1. So this is a figure of a photosystem, and again there's two key parts. There's a reaction center, which is the light blue here, and then there's the light harvesting complexes, which is the purple and green in this figure. So the light shines in and is absorbed by the pigments in the antenna complexes. That energy is then passed between molecules until it hits and is passed to the chlorophyll inside of the reaction center. When that energy is passed to the chlorophyll in the reaction center, you get an excited electron. So that little arrow is the electron getting bounced up to that excited state, and then that electron is taken away by a primary electron acceptor. Again, these are embedded in the thylakoid membrane, and there's two types, photosystem 1 and photosystem 2. So the light reactions actually start with photosystem 2. And so again, there's the light harvesting complexes and the reaction center. The light harvesting complexes will catch the light energy, pass it among the molecules to the reaction center. Inside of the reaction center of PS2, the chlorophyll molecule is a little bit different from PS1, and so we call it the P680 chlorophyll, or P680 for short. So when the light energy is passed to P680, so here's my light energy, P680 gets an excited electron, which is indicated with the little star. So that means there's an excited electron. That electron is then going to leave. So there's the electron getting taken away, and it's going to go to the primary electron acceptor. So remember, that's a redox reaction. Keep seeing those. So now P680 is missing an electron, so we give it a positive sign to show that it's missing the electron. P680 is not going to be able to stay like this, so I'm going to just curve down here. It needs to get back to its original state. So in order to get back to its original state, or really I could draw my arrow like this, right, to get it back to its original state. In order to get back to its original state, it has to gain an electron. This electron is taken away. It's gone now. So a new electron has to come from somewhere else, and this is where water is going to have to be used. So water is going to be broken down to release electrons. That also releases hydrogen ions and oxygen gas. So this is where the oxygen gas is made. But the reason why it's made is to provide electrons to fill the electron hole in P680. So let me walk through this one more time. The light energy shines into the light harvesting complexes. The energy is funneled down to P680, the chlorophyll in the reaction center. When chlorophyll absorbs that light energy, the electron becomes excited. That electron is then taken away or captured by the primary electron acceptor. That leaves P680 missing an electron, so the positive sign indicates that it's missing an electron. In order to fill that electron hole, I'm going to have to get a new electron from somewhere else. That's going to come from water. So water will be broken down into hydrogen ions, oxygen gas, and electrons. these electrons will go in and make P680 plus back into P680, how it started. So at this point, all we've done is move some electrons around. We haven't made ATP or NADPH. And remember that we know that the light reactions are going to produce ATP and NADPH. So how is it going to do that? Well, if I think about the thylakoid membrane, I've got PS2 over here. I've got PS1 here. And in between them, I have an electron transport chain. So what's going to happen is the electron that comes out of PS2 is going to get passed into the electron transport chain and pass down the electron transport chain. And just like in cellular respiration, as an electron passes through the electron transport chain, it's able to pump hydrogen ions from low to high. So that's pumping hydrogens against the gradient. That takes energy. The energy is coming from the electrons moving through the electron transport chain. Still haven't made ATP or NADPH, but guess what? In the thylakoid membrane, I have a protein that you should remember, ATP synthase. So that's our same protein that we've been learning about in cellular respiration. And what does ATP synthase allow? The hydrogen ions flow back through it the other direction. And when it does that, it spins the protein. And as it spins the protein, it's able to carry out the endergonic reaction to produce ATP. So again, the electrons that come out of PS2 that were excited and then taken away from chlorophyll get passed into the electron transport chain. As the electrons move down that electron transport chain, hydrogen ions are pumped against the gradient from low concentration to high concentration with the energy that was in the electron. That gradient is then used by ATP synthase, also in the thylakoid, to produce ATP. So this is the same exact mechanism of ATP production called chemiosmosis that we saw in cellular respiration. Sorry, let me rewrite that for you. Chemiosmosis. Okay. So we're going to leave that electron sitting right here at the bottom of the electron transport chain. And now we're going to talk about PS1, which is also sitting in the thylakoid membrane. So at the same time that all that stuff was happening in PS2 in the electron transport chain, we also had stuff happening in PS1. So in PS1, it also has light harvesting complexes. It also has a reaction center. But in its reaction center, the chlorophyll is called P700. It's slightly different. However, light still shines in. That light energy is captured by the antenna or light harvesting complexes, passed to chlorophyll in the reaction center, and then just like in PS2, the electron becomes excited with that energy. That excited electron is unstable. So P700 then loses an electron. So that electron goes out, and again, that electron is going to go to a primary electron acceptor. And we'll leave that electron there for now. Now again, we need to get P700 plus back to being P700, back to its original state. So I'm going to have to put electrons in there. But remember, I have photosystem 2, I have photosystem 1, oops, sorry, that looks like a 2 there, photosystem 1, and then my electron transport chain. And remember, I have an electron coming down the electron transport chain that just moved from PS2. So this electron is the electron from the electron transport chain, and it's coming down the electron transport chain, and here's PS1 sitting at the bottom of that, ready to catch the electron. So the electron from the electron transport chain will fill the hole in P700. So we can really say that the electron that came from PS2 is filling the hole in PS1. So that's our P700 back to its original state, but we still have this electron floating around to address. So sitting next to PS1 is a protein called NADP plus reductase. And like the name suggests, so this is it written out here if you can't read my handwriting, this is going to reduce NADP plus to NADPH. So remember, that's a redox reaction. This is a reduction. So the electrons that come out of PS1 are going to get passed to NADP plus reductase, and NADP plus reductase will use those electrons to reduce NADP plus to NADPH. So now I've created NADPH at the end of PS1. So in PS1, because I have this electron coming from the electron transport chain, I don't make oxygen and I don't use water. Okay, I don't make ATP, I make NADPH in PS1. So in the photosystems, this is a summary of what's being produced. Oxygen is produced in the thylakoid lumen. Remember, that's the space inside of the thylakoid, when water is broken down to fill the electrons. So this is water being broken down into hydrogen ions, oxygen gas, and electrons. These electrons will fill the hole in P680. ATP is produced in the stroma because of a gradient. The gradient is generated in multiple ways. So remember here, I said this makes a hydrogen ion. That actually increases the hydrogen ion concentration in the thylakoid space. We also said that we have the pump that's powered by the electron transport chain. So this is the hydrogen pump that's going to move hydrogens from low to high. And then the third part, NADP plus 2 electrons plus hydrogen, this uses H plus, so that is going to lower the concentration, so in the stroma. So all three of these things are contributing to the gradient that is then used by ATP synthase. So remember, ATP synthase uses the gradient to make ATP. And the third product, which I already sort of mentioned, was NADPH, which is produced in the stroma in PS1, or rather NADP plus reductase with electrons from PS1. So PS1 passes electrons to NADP plus reductase, which carries out this reaction. So here's a summary figure of that. Here's the light shining in photosystem 2, passes the energy to P680. P680's electron becomes excited and is then captured by a primary electron acceptor. So that gives that electron energy. That energy is then slowly released across an electron transport chain. That energy is used to pump hydrogen ions from low to high. Right? The electron hole in P680 is filled by breaking down water and producing oxygen gas. In the meantime, over in photosystem 1, light has been shining down. It gets passed to P700. It boosts the electron to a primary electron acceptor, which then passes the energy in that electron and the electron itself to NADP plus reductase so that NADPH can be reduced. And then an electron comes from the electron transport chain and lands back down in P700. So you can also think of this as the electron starting here, getting bounced up in energy, so gaining energy, rolling downhill, giving off energy, and getting rebounced up. So this is what this figure is showing. So bouncing it with energy, the energy is being released down here, and then light bounces it back up to energy, and then that energy is put into NADPH. So that's another way to think about the electron movement.